All right. So just to start off with a quick review, of course, I know that many of you on the call have experience with gene curation. So just as a recap, this is the scoring that we use. This is in the SOP. And of course, when scoring case-level variant evidence, the scores are really given within the context of the inheritance pattern of the disorder. So you really need to know the inheritance pattern before starting to score or thinking about scoring for a variant. And in addition to the inheritance pattern, of course, we also score according to the type of variant. So there are different numbers of points given according to the type of variant. And for each type of variant, we have a default score, and then there's also a range of points that can be given. And so this range of points really allows us to take into account the types of evidence that are available that either support the pathogenicity of that variant. So is it weak or strong? So we have this amount of wiggle room here to allow you to score the variant appropriately. So I think this is where a lot of questions come in. You know, what types of variants what types of evidence can I count to support the variant? What types of evidence would make me think I can increase the number of points or may want to decrease the number of points? So that's what we wanted to really try to address today. So, you know, please, as we go through these examples, just let us know if you have any questions or any thoughts on how you would want to score this evidence that we're going to present. So we thought we'd go through each of these sections, you know, sort of one by one. So starting with other variant type. So other variants are typically missense variants, though often we will see small in-frame deletions or insertions can count in this context as well. And this is really directly from the SOP. So there must be at least some impact for the gene function at least some impact to gene function must have been demonstrated for the case to count. So impact based on in silico predictions such as polyphen and SIFT, we would typically score less than the default number of points. And if the impact is based on functional validation, it can score the default number of points or above, depending on the validation quality, the biological representativeness of the functional assay. So really this is when you need to look at the evidence and just really assess it, you know, how strong is this evidence and how high would you want to score these points. Another important thing to think about is this variant rare enough to be causing the condition that you're thinking about? So of course we always look at exact frequencies as well. So starting with a really simple example, ACTN2 is a gene that has been implicated in hypertrophic cardiomyopathy, which is typically an autosomal dominant condition. So this missense variant was found in the ACTN2 gene, and if you look at the pedigree here, there was an adult female who was found to have this missense change. It was passed on to actually both of her children. They're in their teens. One had some evidence of hypertrophic cardiomyopathy, the other did not, but may not have been old enough yet to show the features. This variant was predicted to be probably damaging by polyphen 2, not inexact, so clearly a rare variant, but there was no other information given in this paper to indicate that this variant could be pathogenic or affecting the protein function in any way. So for that reason, we would consider this to be a variant of unknown significance, but not really enough even to give it the default score, because all you have here is the in silico prediction from polyphen. So we would downgrade the score in this case. Of course, the default score is 0.5, but typically in our hypertrophic cardiomyopathy working group, for variants of unknown significance like that, we would give it a fraction of points, so 0.1, 0.2. So any questions about that one, or does anyone have any other thoughts on that? All right, so that was a fairly straightforward one. Moving on to another example from hypertrophic cardiomyopathy, LBB3 is another gene that we looked at, and a missense variant was found in this gene in a conserved CDB domain. Oh, yes.
Jen, sorry, I was trying to get unmuted and it took me a while. Couldn't jump in there.
Okay.
One thing that, you know, just on that last case with the ACTN2 gene would be looking at constraint scores. You didn't mention that in your presentation on that one.
Right. So I should have mentioned that. Yeah. So this is not constrained. This gene was not constrained for missense changes. So that's a good point. That's something.
Because you could imagine a scenario where there was really, really strong constraint against missense variants, where you might actually go to the default, even if there wasn't other functional studies.
Okay. Okay. So that's good to think about too. All right. So that's a good question also for LBB3. And again, I don't recall this being constrained for missense variants. So again, this seemed to be a variant of unknown significance where it's in a conserved domain, predicted damaging by polyphen and SIFT. And in this case, the authors had really done, although in silico studies, a lot of other work to really show that they believed that this variant was pathogenic. So multiple in silico molecular dynamic and free energy studies. I'm by no means an expert in these, but what their conclusion was that this particular variant had significant structural and dynamic impact on the PDZ domain, and therefore they considered it to be a disease-causing mutation. So in this case, and Jonathan, I'd love to get your input on this one as well. I mean, they had done a lot of additional work. I mean, certainly they hadn't done anything like expressing it in cells and looking at what it did, but would this be a case where we might go to the default even though it was only in silico?
I'd be interested to hear what other people think. Any other curators want to take a stab at it?
So I was involved in initially curating this gene, actually, and I remember coming across this variant. This is Bryce Eifert. So the particular papers that were looking at this, and if Kathleen Walls is on the call, I think she also looked at it with the recuration. They didn't just show in silico data, but they did extensive structural modeling, as Jenny's showing there with the molecular dynamic and free energy studies. And there were quite a few people that had looked at this, I think in like 2 separate papers potentially. And so it wasn't just necessarily looking at polyphen and SIF. And so I was almost inclined to give it points just because of the amount of in silico evidence that existed, especially with that modeling. However, I maybe would have given, instead of a default, a little bit less.
Yeah, I mean, my personal feeling is the more work the authors go to demonstrate an effect, the more we ought to give them credit for it. And, it probably gets a little bit, cutting hairs too thin to say, well, I'd give it 0.4 instead of 0.5. I don't think that our system necessarily has that need for that kind of dynamic range in terms of each variant getting, tenths of a point. So I would go with the default on it if they've gone so far as to show it's in a conserved domain. Not only do the usual in silico tools predict that it's damaging, but you also then have additional lines of evidence that would suggest a structural effect. Right. I mean, I don't see any reason why you wouldn't give it the default. That seems like a pretty reasonable, logical line of analysis in my mind.
Okay, great, thanks. All right, so moving on to the next one. This is actually one of Marina's examples. So Marina, I'll let you go ahead and describe this one.
Sure. I just want to thank ahead of time Sarah and Brandon, who are curators in the hearing loss group, for assembling pretty much a lot of these curations. I've pulled a lot of the examples from that, so shout out to them. So this is DNMT1, which is associated with a syndromic hearing loss condition, DNMT1 methylopathy. And this variant's a missense variant. They had done a mixture of computational predictions, so you can see on the bottom right that it's in a highly conserved region, and a lot of other species have the same variant, or the same amino acids. But then they also transfected it into the variant into HeLa cells and found that the wild type variant is localized to the nucleus, and that the missense variant causes mislocalization from the nucleus to the cytoplasm. So you could argue potentially that the GFP tag is altering localization, but in this case, we thought with a combination of the in silico conservation prediction and also that functional study that it was valid to give it the default score of 0.5 points, and then also to score that separately as experimental evidence.
Were you scoring the experimental evidence in favor of the gene disease association or in favor of the variant itself?
The gene disease association.
Huh. Okay. I mean, I guess my normal intuition would be if you're showing an effect of a particular mutation on some, you know, it's hard to know exactly when to count it for gene level evidence versus variant level evidence. I mean,
it's tough. It's generally hair splitting, but I agree with you, Jonathan. Usually, if it shows that the variant is altering splicing or localization, like particularly variant level, that we would just upgrade or downgrade the variant score.
Yeah, I would have probably given it. I'd probably give it more points on the variant level than the gene level evidence, but.
Yeah, that makes sense.
What do others think? I mean, this is a really challenging part of the framework, is when do you put functional evidence in what we call the gene level evidence, and when do you put functional evidence in what we call the variant level evidence? You know, this is a pretty clear example of a variant that has a lot of evidence supporting it as a, you know, damaging variant based on evolution, based on, I presume that it's rare. We don't see that here, but one presumes that it's novel or rare. And you get this mislocalization due to this one mutation. I'd probably call it at least 1 point per variant.
This is Brandon. I was the curator who curated this gene disease pair. Wouldn't you also count the, you can see the pedigree on the right here, wouldn't you also count that segregation as kind of evidence towards this variant being pathogenic?
So talk us through the pedigree and who has what.
So if I remember correctly, although I didn't put it here, there were no non-segregations in this. The black ones all did have the condition and the variant.
And that's,
right, so,
that's a great question.
You would count that towards pedigree data then?
Yeah, I agree.
So I don't think that you would want to count it both towards increasing the weight of evidence for this specific variant and for the linkage or pedigree data. So you'd probably calculate or estimate a LOD score based on your segregations, and also, you know, take the variant and its information and score it for itself.
Right. So in this case, I was just using that as evidence that it should receive at least just the default. And then in addition to that, counting the segregation separately, the points.
Yeah, I mean, I could imagine a situation where you would see some segregation but not feel confident in calculating a LOD score for some reason or another, but you'd still want to acknowledge that it looked like there was, you know, reasonable evidence of segregation to allow the variant to have some default points. I think if you're counting a calculated LOD score, that probably shouldn't be double counted in the variant, right? Because you've already applied that line of evidence in the pedigree LOD score estimated calculation.
Yeah, I agree. I don't think I would ever use a pedigree to upgrade a variant score.
Right.
Just double counting.
So, Jonathan, are you saying that you could imagine a case in which you would give a variant points for segregation, but also give it, and let's say it doesn't have any functional evidence supporting it, you would give the variant less than the default, but also still count the segregation points separately?
I'm not sure I understand your question. So I think what you have to do is you have to separate those two lines of evidence, where what we're really saying with segregation is that there is evidence of linkage of this region, and in this, you know, it's never really the specific variant itself. It's anything that could be in linkage disequilibrium with that variant. So the segregation evidence is really about, are we convinced that this, you know, this tiny region of DNA contains a pathogenic variant? Whether or not it's this specific one, it could be a, you know, an intronic splice enhancer variant that you didn't pick up by exome sequencing, but is actually the pathogenic variant, and this missense is in, it's just in LD with it. So, so you'd sort of would, that's why we created the separate pedigree LOD score calculation for segregation evidence to give you the ability to score just the segregation in that one context. So you could have a variant that was pretty flimsy on the functional side. Maybe it was a missense variant. Maybe polyphen and CIF disagreed, or there was very, very minimal evidence of pathogenicity of the variant itself, and you could give that variant a very low score, but count the fact that there was good segregation that would go in favor of the gene disease association. Does that make sense?
Yeah, that was perfect. That was exactly what I was looking for. Thanks.
All right. So, any other comments or thoughts on this example?
I think we really do need to address the question of the functional evidence and when to apply it to the variant and when to apply it to the gene, and that's probably something that further work on the gene creation working group, we could sort of bring this back to the group and sort of get further guidance on it.
Yeah, absolutely. So I think, as you know, Jonathan, we have a lot of questions about this too. So I was almost thinking about getting something together where we kind of have specific types of evidence and show when that might count towards the variant and when towards the experimental evidence. So I think that would be hopefully useful.
Hey, Jenny, I have two questions. One is, and maybe I missed this on the segregation call, but can you count segregation when they don't show that it's segregated in another individual who's affected? I mean, it looks like, if I'm reading this pedigree right, they just looked at unaffected people and showed they didn't have the variant, which to me doesn't mean that the people that are affected have it. And two, I'm wondering, and maybe this is an example coming up, do you have an example where you would give something a 1.5, or can someone give an example of when they would give a 1.5?
Possibly, yeah. So there's definitely some examples coming up which have a lot of evidence to support them, and I think some of these you could potentially give 1.5. But your first question, so you were saying that in a pedigree like this, if they're only showing that the variant is present in the affected individuals and haven't looked in the unaffected to show it's not there, is that what you're asking?
No, the opposite. If I'm reading this right, it looks like, I don't know if the people that were genotyped are the ones with the asterisks, but if so, then they only looked at the proband and then they looked at unaffected people. It doesn't look like they confirmed that any of the other affected people had that variant. Maybe I'm misreading it. But whether I'm misreading it or not, I mean, would you count segregation points if they didn't show it was the variant was in another affected individual?
You need to, they need to have genotyped those people in order to count them for segregation. So that's my question at the very beginning of this was, I can't tell from this pedigree who was genotyped and who wasn't because they didn't label it for us. And so, but the people curating it said that they were genotyped. Is that correct?
Okay, that's for the asterisk and something else. Okay, thanks.
Okay, so next example, my screen. There we go. So this is another autosomal dominant example for oligodontia, the LRP6 gene. So this variant was identified in this family with clear autosomal dominant inheritance, so that the red boxes indicate those individuals who were genotyped and had this particular missense change. And of course, the solid black indicates that they are affected by oligodontia. The number here shows the number of missing teeth. So, Christy, you were asking for examples where you might actually count 1.5. I'm not sure you go up to 1.5 on this one, but I would definitely, I think, upgrade the points. What they showed for this particular variant is, let me backtrack. So LRP6 is, it encodes a transmembrane cell surface protein, and it actually functions as a co-receptor in the Wnt signaling pathway. So in order to perform its function, obviously it has to get to the cell surface. And so when they found this variant, they found that it was predicted to affect the cleavage of the signal sequence. And then for some reason, I'm not sure why they looked at the mouse protein, but they were able to generate the equivalent missense change in the mouse protein, and they expressed it in HEK293 cells. And what they found was, you can see in this figure here, this is basically a Western blot, and they showed that the variant resulted in lower levels of the protein and also that it was immature. So this is the wild type, so you can see the lower band here is the immature form, whereas the band with higher molecular weight is a glycosylated form. So they actually did a bunch of different experiments that all led to the same conclusion, that the mutant protein was retained in the ER due to failure of cleavage of the signal peptide, so it wasn't able to reach the plasma membrane where it normally functions as a receptor. And they showed that indeed in these cells that were expressing the mutant protein, there was loss of function of the receptor. So multiple lines of evidence that were all kind of telling the story about how this particular missense change was affecting the function of the protein. So I would definitely be comfortable upgrading points. I think 1 maybe, I mean, because it's a mouse variant, I don't know. I mean, definitely 1. I don't know if anyone would have maybe scored it higher. Any thoughts on that? There's no gene constraint for this one for missense changes, and but any other thoughts on that?
There is no. I thought there was constraint looking at it in exec.
Oh, is there? Okay. But for so that's another question actually. When you have, so for oligodontia, you wouldn't expect that to be a condition that's going to.
It's true.
Yeah, so that's another question. I think this came up for one of our cardiac genes too. I forget which one I was looking at, but there was very little evidence to support its association with HCM, but yet it was known to be involved in one of the arrhythmias, and it was highly constrained. But then I wasn't sure how I could use that information for HCM because perhaps all the gene constraint was due to this other arrhythmia. So that's right. Another thing that we can sort of think about.
Definitely should always be on your mind when you're thinking about the constraint, for sure. Okay. I mean, I think that this is functional evidence that the authors have gone to almost heroic measures to show that there is a deficiency, that this actual variant is creating an abnormal protein.
Yeah,
right?
I agree. It was really nice. Yeah, they've done a bunch of different things.
I don't see why you wouldn't go to 1.5. I mean, I don't know what else. What else would you want other than to make a mouse model of this specific mutation that caused oligodontia? I don't know.
Right.
Right. I think it's pretty clear that the variant is damaging.
Okay. That's great.
What do other people think?
And Jonathan, I'm glad you brought up the mouse models, because I think that's another question that we've had. I mean, we've typically been counting mouse models towards the gene-disease association because you're really, if there's recapitulation of the phenotype, you're really showing the association between variants in the gene and kind of the phenotypic effect. But, I mean, I guess, would you agree with that?
Yeah, because the mouse models are going to give you more points anyway because the default amounts that you could gain for it, and so you're going to want to put it in that category. But this is definitely a clear example where functional evidence here has more to do with the variant than the disease, right? Because what they're showing is abnormal cleavage, abnormal retention, blah blah blah. So that clearly needs to be counted here and not in the gene level functional evidence.
Absolutely.
The previous example where the mutation keeps it out of the nucleus, you could argue, well, maybe that's part of the disease mechanism because the DNMT is supposed to be a methyltransferase, and so it can't act on the DNA if it's not in the nucleus. So you could sort of stretch the argument and say that that would be gene-level functional evidence. But in this case, I think you'd have to count it as variant-level evidence.
Right. So that's a really nice distinction to make, I think. And I think that's why I was comfortable to counting this for the variant rather than towards the disease, because, like you say, there's not really any link between the abnormal function of this protein and the condition. So, all right. Okay, so
I did have a question, Jenny, before you went on. This is
Sure.
Courtney. Going back and saying with this variant, and you had brought up animal models, and we had talked about, you know, because they can get more points counting them for experimental, but I have had a few cases where I've had multiple mouse models made independently that are overexpressing a specific variant. Would I then count both of those mouse models towards animal, or can I take one of those and count them towards variant points to increase the variant?
Yeah, that's a good question too. How, you know, if you have an overabundance of a certain amount of evidence, can you kind of, instead of counting it in one area, count it in a different area? So any, I don't know, any thoughts on that? I mean, often I guess these genes, was this a definitive gene, Courtney, or?
It was a definitive gene, so
Yeah.
I mean, it's probably a mute point, but you know, on some of them I have seen where they will start to get into multiple animal models. So I wasn't sure how we could do that.
Yeah, that's a great question. Jonathan, do you have thoughts on that when you have kind of an overabundance of evidence?
Right.
You know, can you count?
Yeah, I mean, yeah, exactly. When it's definitive, you know, it is definitive. It's, you know, we don't worry so much about over calling things, and I think that the main danger is that you want to try to we want to be equal across different gene disease pairs in how we're treating the evidence, so that we don't have sort of heterogeneity.
Yeah.
You know, I don't think it would be wrong to say that for a particular missense model system, you know, that you applied it to the variant because you felt like it really supported the variant effect, and in another setting, if it was a different model, applied it to the gene. I mean, it is a hard call. I think since there were multiple models, then you ought to give give as much credit where you can, kind of thing.
Right.
Okay, thank you for that.
I have a question. This is Calotina from LMM. Regarding the LRP6 variant, I have trouble understanding why give it the maximum points, because unless, I mean, they do something completely in vitro, and they chose a cell line that, although I'm not familiar with the disease, there's no rationale between the cell line that they chose to do their experiments and this particular gene. Yes, so I would feel more comfortable to give them more points if they had reproduced what they see in this cell line in another cell line that was more relevant to the disease, maybe. So I agree they have done a lot of experiments, but maybe they could have done more.
Yeah, and perhaps sorry, go ahead.
Because we all know that what we see in vitro doesn't necessarily reflect the reality in the system that is relevant to the disease.
So perhaps if they had used a system more relevant to the disease and could really show how it was related to the phenotype, that might have been more on the experimental evidence side of things. Whereas, you know, here, I guess they're clearly showing that the variant disrupts the function of the protein.
In this cell line.
Yeah, in the cell line, exactly.
I mean, that's the only reserve that I would have, that they show it in one cell line.
Right.
And without having read the paper, I don't know why they chose that cell line.
Yeah, I don't think they stated exactly why. I mean, I guess HEK293 cells are kind of commonly used. And why they use the mouse variant and not the human. Yeah, I mean, I don't know. Having read the paper, you know, they really did they did multiple experiments that all kind of led to the same conclusion, that the appropriate targeting of the mutant protein was was disrupted, and it couldn't get out to the cell membrane, couldn't do what it was supposed to. So, I mean, clearly the function was altered in this particular cell type, but I don't know. Anyone have any other thoughts?
Hi, Jenny. This is Courtney again. I agree with the assessment of seeing that it's in HEK293 in mouse, but I also agree that instead of being more experimental, it should be more towards the variant, because while it isn't towards the disease, it is still showing a function. And because that range can't give you as much points as if you did experimental, maybe it's more appropriate to be put in here, is that you want to give a little bit of extra points, and this gives you that wiggle room without overinflating the experiment that was done.
Right. I guess the question is, oh, sorry, Jonathan.
No, go ahead.
I guess, yeah, so I guess the question was, should we really be giving it 1.5, or would you be more likely to give it 1,
So here's, clearly trade-offs, right? And I think that our framework has enough dynamic range that you can, that extra 0.5 is probably not going to change your final result. It would be really interesting to know what the final score was and whether 0.5 points was going to change that. My sense is it's probably not. I think we probably spend too much time being overly critical of variants because we're thinking about them in terms of pathogenic or likely pathogenic sometimes. And that's not the point of the scoring, right? The scoring is about, is the preponderance of the evidence in favor of this gene-disease association? And look, somebody's graduate student spent 9 months trying to get that page worth of figures done, right? If you look at that figure 2, there's 4 or 5 panels. Somebody spent a whole lot of time doing that, and we need to give them credit for it. I think we get a little bit hypercritical of this stuff.
Yeah, I completely agree with that. Is this pathogenic or not? But yeah, absolutely. Just looking at the weight of the evidence and yeah, giving it its fair scoring. Yep. Okay. All right. So those were all the examples that we had for autosomal dominant, sort of other variant type, mainly being missense. So moving on to null variants. So here, when we're thinking about null variants, we're basically thinking about variants where no protein is produced. So of course, these are typically nonsense, frame shift, canonical +1, -1 or 2 splice sites, initiator codon, single or multi-exon deletions. And of course, there is not every single one of these type of mutation is going to be a null. Of course, we need to think about nonsense-mediated decay. Sometimes a splice site may skip an exon, but the protein could still be in frame. So you really have to think about these things too. But in general, these types of variants are considered very strong evidence for pathogenicity in genes for which loss of function is a known disease mechanism. So again, that may come out of your literature review. And it can be tricky, I think, for genes where there's very little information available. I was creating a gene for HCM recently, and it was an extremely limited gene. I think we ended up giving it 1 point or something like that. And there was a nonsense variant in there, but because we didn't know that haploinsufficiency of this gene could cause HCM, we just counted it as a variant of unknown significance and gave it very minimal points. So these are really all things that you need to think about. But the disease mechanism can be assumed to be loss of function if the gene is loss of function constrained. So of course, that's something that you need to take into consideration. Loss of function constraint scores should be interpreted in the context of the disease in question. So if it's a condition that tends to have later onset in life, after someone may have already had their children, there'd be less chance of seeing gene constraint versus a severe pediatric onset disorder. And you can assign more points if the gene is known to be highly constrained for loss of function variants. And as I mentioned, of course, thinking about things like alternative splicing, or if a premature termination codon is near the C terminus and nonsense-mediated decay wouldn't be predicted. So these are all things that you should consider when looking at null variants. So one example of this, again for LRP6, autosomal dominant oligodontia. There was no additional experimental evidence provided in this case, and they found these 3 different frame shift variants, which were inherited in an autosomal dominant manner in this family. And I would have just given these the default score for these. The next one, Marina, do you want to talk about this one?
So this is autosomal dominant since we're a neural hearing loss. KIT LG, it's a ligand. So I realized actually that I have the wrong variant bolded. It's actually the one that has boxes around it for the functional evidence. It's a truncating variant, SARE96TER. So they had a couple, so you see the pedigree, but then they also had a couple pieces of functional evidence. The first one showing that in transfected 3T3 cells that the 28 kilodalton band for KIT LG is gone in the cells transfected with the variant. And then also, as I mentioned, it's a ligand, so it's secreted. And so the wild type is secreted when you measure via ELISA, but the variant transfected cells don't secrete any protein at all. So I had suggested to upgrade in this case the scoring points for that, maybe up to 2 points potentially.
Right. So any other thoughts on that one? Right. So moving on to de novo variants. For de novo variants, points can be adjusted depending on the statistical expectation of de novo variation in the gene in question. Both parents must be tested to show that the variant truly is de novo. And of course, just looking for the variant and if a parent doesn't tell you that the parents truly are the parents, especially with paternity, so the score can be increased if maternity and paternity are confirmed. Excuse me. So this is an example where the PDE10A gene was sequenced in patients with an early onset form of chorea. I think it was very early childhood onset chorea. And there were missense changes identified in 3 different families, actually. So this was one of the families, here's another, and the third family is on the next slide. These were identified by whole exome sequencing trio analysis. Both of these missense changes affect highly conserved amino acids, and they were predicted by in silico modeling to have an essential role in the CAMP binding of this phosphodiesterase protein. And they also did some work where they had showed that they had expressed these different variants, again, in HEK293 cells, and showed that there was an activation of the phosphodiesterase activity by cyclic AMP was affected by these particular variants. So this is the wild type, and this is the mutant cells, and you can see that the hydrolysis of cyclic AMP was altered. So they have certainly the functional evidence, they have in silico evidence as well. They show in the paper that both of the parents were wild type. So a question that we've actually been discussing here as well, they had done trio analysis by West, and what they state in the paper is that, I'm going to say that was what they read, the family-based sequencing approach of these individuals directly indicated that both mutations had occurred de novo. So my assumption from that is that they had really looked at the variants in the parents and the affected children and could really, from that, state that these really are the parents, but they don't, that's as much as they say. So we have had this question about trio analysis and how, can we say that it's confirming paternity, or what do we look for in the paper to kind of say that, yes, we really feel that these truly are the parents? I mean, it seems like they were making that statement in this particular paper, but any thoughts on that and scoring for this one? I mean, I feel like we have some experimental evidence. We have evidence that these really truly are the parents. So I probably would have given this higher than the default, maybe as high as 3. But thoughts on that?
Yeah, I think if they did trios, then it's pretty unambiguous that the parents are biologically parents. Otherwise, there would be, tons and tons of variants that would have showed up as not following Mendelian segregation that would have been flagged as an error of some kind. So I think you can be pretty certain that their paternity is confirmed here.
Fantastic. That's great. Thank you. So any other thoughts on that one? All right. So this was the other family I was talking about where they had also found this particular missense change. And I thought this was interesting because in this case, you can see it's a large SIB chip, but the parents are deceased. So what they did was a haplotype analysis where they looked at all of the siblings and they were able to identify what the parental haplotypes were. So here you can see they've denoted them with different colors. There's red, green, yellow, blue. And the affected individual here has the red and yellow haplotypes, and the unaffected SIB has the same haplotypes. So from that, they basically said, they were pretty confident that this was a de novo variant. So, I mean, I think I agreed with that, looking at the different markers that were, flanking within the locus. So I thought that was interesting, and I would have, I think, been comfortable assuming that this truly was de novo based on this. Any other thoughts there?
Yeah, I think that's right. I think you can't formally exclude the possibility that one of the parents was a mosaic.
Mm-hmm. Right.
But, I think it's pretty reasonable. At least,
okay.
It certainly doesn't appear that that the parent with either the red or the yellow haplotype that the variant is on, that it wasn't a full germline mutation because you've got another SIB who had the same haplotype without the mutation. So it would have had to have been sort of a mosaic of some kind, if other than a true de novo.
Right, and I mean, I guess that, that can happen too with, if you're only looking at lymphocytes for even West or paternity testing. I suppose you've always got that possibility, right? If, how do we tell something is truly de novo? Yeah, okay. All right, so this is another example of a de novo null. This was an early onset generalized dystonia where they had found de novo splice site change, and what they did here, in this case, they had found this by West, but only West of the individual, and then they had Sanger sequenced the parents to show that they did not have that same change. So in this case, it appears to be de novo, but they did not confirm paternity. What they did do was to go on and show that this particular variant does affect splicing, as you would expect. It's a -2, of course, except for splice site change. But they did show that looking at cDNA. So basically, they found that there was skipping of exon 29 and then use of a cryptic acceptor splice site in intron 29, which results in this abnormally sized fragment. And that results in a frame shift. So they did show that it affects splicing, and there was also reduced expression of the protein in patient fibroblasts. So here, I may have given this the default score. I mean, I guess because they didn't confirm paternity, I felt like I couldn't really go any higher than that. Any thoughts on that? So, all right.
It seems reasonable.
Okay. So another question that's come up for us recently, we have this, various questions I wanted to present, because I feel like if we've had these questions, maybe other people have too. So what are acceptable assays for genotyping the parents? And this came out of a paper that Courtney had looked at recently, an old paper where they'd used heteroduplex analysis to first identify variants in patients with tuberous sclerosis. And you can see clearly there's a different pattern here in the affected individual versus the parents. So I think I would be pretty comfortable, counting that. I mean, it's not the gold standard sequencing, but what do y'all think about that? And I guess sometimes we see SSCP shift, heteroduplex, other RFLP analysis, other things that you might use to look for a particular variant in a patient that aren't, they're not Sanger sequencing, but it still shows that the variant is either present or absent. So any thoughts on that?
Yeah, I mean, it all sort of boils down to what is the, what's the sensitivity of that assay to detect, like, again, getting back to the issue of mosaicism. I mean, that blot right there looks pretty,
it looks pretty clear cut.
Or electropherogram, I guess, is probably what it is. But in any case, yeah, it looks really clean. And so,
Right.
I'd be inclined to believe it.
Okay.
And again, this
Okay.
is one where, you know, you guys are not all supposed to be experts on every possible molecular assay, and we do have to trust the authors of these papers and the fact that they've spent their lives working on these, you know, these conditions, and that they pass peer review and so forth. And so rather than, you know, having somebody go off and now let me go and find out what would be the, you know, limitations of a heteroduplex mobility shift, you know, that was pretty much the standard at the time, and that passed muster, and we just say, you know, default.
Okay.
Yep.
Right. Okay, that's really helpful, because, you know, we sometimes see this for just a, you know, regular family analysis too, you know, who had the variant, who didn't, SSCP, heteroduplex, that type of thing. So, but yeah, I mean, this is, you know, a really nice gel. So,
I do have another question to that effect with the going through the authors and what they stated, because going through some of these older papers, you will not have the nice, you know, electropherogram or diagram showing the actual mutation, but you will have a list of patients and whether they were familial or sporadic. Would we still be able to count those cases where they're saying sporadic if they are not actually showing you the data in which they've interpreted?
That's a good question.
I guess I've supposed in the past, if I've read in the materials and methods that all the patients and their parents have been tested, then I have been inclined to count the sporadic. But if I can't find that assertion made, I typically don't count the sporadic cases because I don't know that the parents were genotyped.
Right. So if they don't give further details on what exactly they did, but they just state that patient was a sporadic case, but you don't know how that decision was arrived at, then you'd be less comfortable counting that.
Exactly.
Yeah, and sporadic is probably an old, outdated term that we probably shouldn't use, and simplex is the better word. And the assumption is that, you know, that's the only person in the family that's known to have the disease, right? But without phenotyping other people in the family, could you exclude the possibility that somebody had sort of mild? Yeah, so you do have to sort of go with whatever they say was done in their method. And if they said that they did genotyping of all, you know, individuals and their parents to show that these were de novo, then you could sort of take them at their word, or that they phenotyped all of the family members, et cetera, that kind of thing.
Okay, thank you.
So, all right, so we were going to start talking about autosomal recessive conditions as well. We have just a few minutes left, so maybe we'll start this section and then we can continue with the examples on another call. So, Marina, do you want to talk about this one?
Sure. Yeah, I think this is a discussion that can be outside of the variant scoring anyway, and I just wanted to talk about it because it was a discussion we had within the gene curation small working group before. Is if you notice that you look at the genetic evidence matrix, pretty much all of the variants for autosomal recessive say 2 variants in trans. So essentially confirming that variants are in trans. And so at least the discussion we had with the gene curation small working group was that if a proband had homozygous variants or was homozygous for one variant, that obviously you wouldn't need to confirm that it was in trans to give the scoring. But if a proband was a compound het, you would need to see some sort of confirmation that these variants were in trans to give scoring for the variants. So we could definitely talk about what sort of confirmation that would be. Usually we're just looking for some sort of sequencing or pedigree analysis for the parents or potentially other relatives to confirm that maybe one parent has one and one parent has the other, and then the child has both, that those variants would be in trans.
Okay. So maybe we have time for one example before the end of the call. Marina, this is yours too?
Yeah.
Do you want me to show the figure too?
Yes. Sorry. So this is an example where confirming that 2 variants are in trans is kind of important, because if you just looked at the individual in isolation, you see that any of the individuals you would point to has 2 missense variants. However, if you look at the pedigree analysis, the two missense variants are actually in cis, and affected individuals have 2 copies of the 2 in cis variants to be affected. And so I think this is one of the examples that points that you should confirm indeed whether the variants are in trans before just blindly scoring and assuming that they are in trans, because these are very close together, so likely they're linked, but variants could potentially be in cis, and I guess one warning would potentially be their position on the chromosome. Are they very close together? But oftentimes for autosomal recessive, you should confirm that they are in trans.
So this actually poses an interesting challenge to the curation interface, because we're entering the variants separately, whereas what you actually have here is essentially a haplotype, or an allele that's composed of 2 different changes. So what would you say, I mean, how would you score this? Because either one of those variants could be the causal one, or neither.
Yes, that's correct.
So what was your take on scoring this, the sort of combination of variants?
Yeah, I think in our case we essentially scored them as a haplotype, where they were so linked that you couldn't tell which variant was which, and scored them as one variant almost. But I did have difficulty putting this into the interface, and this is something that I feel like we should discuss for sure. Yeah.
I mean, you could imagine a situation where, further work is done on both of these variants, and one of them is super common, and you could say that's probably benign, and this other one is rare, and it's got some in silico evidence, so that's probably the one that we should score. But these types of situations will come up from time to time, and it's worth thinking about, how we would score it.
Right, especially if there's no additional information. They haven't taken each one separately and expressed them in cells or something like that. Right. It's really hard. So
because if nothing further has been done on either of these variants, then you might only want to count this for segregation data.
Yeah, I agree.
Right, unless there's something else about one of those variants that makes you think that it's worth giving it a default for missense.